Okay, so we're going to go over chapters 1 and chapter 3. Chapter 2 is like the molecules of life. And so definitely review that chapter, but there won't be any questions specifically about that chapter in the exam. Later on in the semester, when we're talking about cell structures, we might be talking about what the structures are made up of, carbohydrates, proteins, and that. And so knowing those terms are important. So with chapter 1, this introduces you to microbiology and kind of gives you an overview of some of the topics we'll be talking about this semester. So there's a lot more information in your textbook or whatever textbook you're using for this chapter than what we're going to be covering. I'm just picking out really key pieces to introduce you. For chapter 3, this is like a techniques chapter, so kind of giving an overview of techniques that you'll be doing during this semester. Okay, so when we're looking at microbiology, we want to have a working definition. And so a good definition of microbiology is the study of living things that are too small to be seen without magnification. So these are too small to see with our naked eye. We're going to have to use some kind of microscope or instrument in order to be able to see them. A key part of this definition is living things. There are some exceptions to that, and we'll be talking about that in just a moment. So just keep that in mind. Like two of the really important parts of this definition is living, and that you need to have magnification. Okay. So when we're looking at microbiology, we're looking at microorganisms or microbes. Sometimes they're commonly called germs, viruses, bacteria. Sometimes in lay people articles or newspapers and magazines, they interchangeably use viruses and bacteria. And so we just want to make sure that we're conscious if we're using the correct term. And so instead of calling these germs or bugs, microbes would be much preferred when it comes to like exams or anything that you're passing in. Okay. We do want to keep in mind that not all microbes cause disease. If we think about it, everyone's been sick at some point in their life, most likely, either whether it was a cold or flu. But the majority of your days, you've been healthy. But you have lived with microbes every single day of your life, from birth, and really even before birth, but from birth on, you've been exposed to microbes. And again, for most of us, most days, we're extremely healthy. And so they're really essential to human life. And there's been increased research being done through the Human Microbiome Project, and we'll talk about that during Unit 3 when we're talking about microbe-human interaction, looking at not just, disease, but really about what beneficial things microbes do, everything from metabolism to development of your immune system. So they're very important. So we want to keep in mind that we don't always want to kill them. So there are several different types of microorganisms. So bacteria, algae, protozoas, hemolyths, and fungi. So hemolyths are parasitic worms, and so in our department we have a course called Parasitology. That's Bio 316. So it's offered both in the spring and also summer session for any of our bio majors. So with hemolyths parasitic worms, these are pretty large, and so at some point of their life cycles, you can see them without the naked eye. If you've ever had a pet that's had worms, you didn't need to get a microscope out to see them. Now, the veterinarian might actually take a fecal sample and do a smear and look at that under the microscope to see them during their cyst or egg phase. But again, so keep in mind the definition. Hemolyths don't quite completely fit that, but they're gross. And so a lot of gross things get covered in microbiology. With protozoas, this is another one that's kind of focused in the parasitology course, so we won't talk too much about them this semester, but these are unicellular organisms that, and for the most part, are motile. There is a group of them for classification that are non-motile. But again, we tend to focus those more in the parasitology class, but we might touch upon them because there's some really cool ones like Giardia that you might have heard about. And these two have like a cyst phase or like an egg phase, like a dormant phase, just like with the hemolyths. So then we have are another eukaryotic group, the algae. And so for algae, the only real disease we have is red tide. So for this course, we don't usually talk too much about algae. If we were looking evolutionary, they're interesting for the course, but for our purposes, because they're really limited to more of ecosystems or food. So some people consume algae in their diet, part of their diet, and so they would be relevant in a food microbiology class or an environmental microbiology class. So usually we don't have much time in the course of the semester to touch on either. Maybe we'll bring algae in when we're talking about the articles at the end of the semester. So that leaves us with bacteria and fungi. So fungi, those are a eukaryotic organism. They have a cell wall, and we will be looking at those this semester. So we'll talk about antifungals when we talk about ways to kill microorganisms in our second unit. Keep in mind we are eukaryotic, so we are more like fungi than we are bacteria. Now, bacteria are prokaryotic organisms, so these are sometimes thought as simpler than eukaryotic organisms, but they have unique pathways and unique functions. Traditionally, in most intro-bio courses, we focus more on cell structures or organelles that are found in eukaryotic organisms, and prokaryotes are kind of like, we'll worry about those later on. Well, we're going to worry about them now. So most of what you'll be doing in the lab will be with bacteria. You might end up growing some fungi, but you should be looking to isolate bacteria in the lab. And most of what we'll be talking about throughout the semester will be focused on bacteria. So they really are dominant microorganism for the semester. Lastly, we have viruses, and viruses are non-cellular. So all the other organisms are made up of or have a cell or a collection of cells. With viruses, they are not a cell. Okay, so they're not able to self-replicate, which is a key part of being alive. So our other microorganisms are able to self-replicate, so they can grow and divide, grow and divide, grow and divide. And they can either protect you or they can make you sick. Viruses cannot self-replicate. They actually need to use a host. So they really are parasites because they're going to feed off, use the host machinery inside the cell. So they're intercellular. They go inside a cell, the host cell. They use the ER, the ribosomes, and they make copies of all of the viral proteins and genomic information. And then they package it back up into new viruses, and then they either lyse the cell or they bud off the cell, but they end up damaging the cell quite a bit to the point that the cell in some cases dies. And this is where we can have cytopathic effects to the cell. So we can actually look at tissue underneath the microscope, and it looks different. Those cells, the host cells, look different, and that lets us know that a virus has been infected them. Okay, so we will talk about viruses this semester, but again, bacteria are going to be our focus. So when we look at microorganisms and microbiology, we really can look at this evolutionary timeline. You know, humans were just really a blip on the radar of the life timeline on Earth. And if we look backwards, we go from humans, you know, we're pretty complex mammals. You know, before us there was mammals, and then there was other organisms that were multicellular with tissues and such. And before that, you know, about 2 billion years ago, the first eukaryotic appeared. Before that, over 3.5 billion years ago, we had the first prokaryotic appearing. As I mentioned, prokaryotics are a little more simpler. They don't have the organelles, they just have these cell structures inside. So there's not all this packaging and membranes. So next week when we talk about cell structures, we'll talk about some of this packaging that happens, like with the ER. Well, prokaryotes don't have ERs. So they're really just like bare minimum. What do you need to get the job done? Let's put the enzyme just in the cytoplasm kind of thing. With our eukaryotes, there's some bells and whistles, and that allows them to start to specialize, right? So we see that further specialization when we get the tissues and organ systems that we see in more complex organisms like us. So it's important to keep in mind with prokaryotes, each individual cell is its own organism. They might work together as a collection to do something, but they're still their own individual. And even those early eukaryotes, just like when we look at our protozoas are unicellular. Each one of them is their own individual. They might share the same, they might be clones, so they share the same genome, the same genetic information, but each one is acting by itself independently. Again, they can work together collaboratively, but that's usually to their own benefit. Now, when we look at humans and microbes, there can be beneficial relationships, and I hope you do walk away from the semester appreciating some of those beneficial relationships. But there's also the pathogenic, and so a lot of times, either us in society or even like with nursing students, you guys are doing clinicals and you're seeing all these people getting sick from different infections and everything. So we sometimes focus on the pathogenic ones more so. And this table's from the textbook that I had for supposed to be at the bookstore. And you'll notice that some of these diseases are in pink. So like influenza and pneumonia, septic, HIV and AIDS. And these are all clearly caused by a microorganism. And so we definitely have, when we have these interactions with microbes, sometimes they're pathogenic. The one thing with this table is we want to keep in mind that even though some of these are not in pink, there is at least a subset that is potentially caused by pathogens or microbes. So we're seeing increasing evidence that some cancers are caused by microorganisms. There's also some different heart disease, subset of heart disease that can be caused by microorganisms. And there's theories that things like Alzheimer's is caused potentially by a microorganism. Diabetes, looking at type 1 diabetes, there's evidence that Coxsackie virus, this virus that goes to the pancreas, actually can initiate individuals to have type 1 diabetes. There's also evidence that the microbes that inhabit your GI tract affect your metabolism. So potentially they can be evidence is supporting that if you have a shift in your gut microbes or certain phenotypes or certain presence of microbes present, you might be more susceptible to developing like type 2 diabetes. So I do think in future editions we'll see that more of these, maybe the authors of the book, the textbook, will have another shade, so they'll have yellow or something, and those ones will be the ones that are like a subset is caused by microorganisms. Who knows? I don't review this text, so maybe I should make that suggestion. With our beneficial relationships, we have that normal interaction that we have with microbes, and they protect us, they help our immune system develop, they help with nutrition. But on a kind of different thinking outside of us as the host, we can also use microorganisms to engineer or to synthesize compounds that we can't produce in the lab. So one area that this is utilized is with type 1 diabetics. So type 1 diabetics would have to inject themselves with insulin. This insulin used to be isolated by cows, but that became dangerous because of mad cow disease. So then they were using pig insulin. Pig insulin wasn't regulating blood sugars as well, so we're having people that previously with cow insulin was really regulated. Pig insulin, they were having lots of highs and lows, so that's not good for them and their overall outcomes. So what they did was they took the gene for insulin, they made the necessary modification because there's splicing that happens to insulin, and they produce that in E. coli. So they can have E. coli producing insulin. They then purify the insulin out, package, ship to the pharmacy, and now type 1 diabetics will use recombinant insulin in order to maintain there. And they can further modify that recombinant insulin so you can have short release and long release and everything. So a lot of these biologic drugs, so if you've listened to commercials and you hear about biologics, most of those would be genetically engineered in microorganisms, and E. coli is a pretty common one. Lastly, we have bioremediation. This is where we could use microbes to clean up an ecosystem. So if an ecosystem's been damaged, usually because of human interactions, we can release microbes that can actually help with that. So, if there's an oil spill, there are bacteria that can actually consume the oil. So they would be able to consume the oil, and that would help to bring that ecosystem back to its more natural form. More recently in the news, there's microorganisms that can eat plastic, right? So we have all this plastic waste that's polluting the earth. And so how do we get rid of it? Because it's going to take not millions, but hundreds of years for it to break down. Well, there's a microorganism, there's a bacteria that can actually eat that plastic. So it utilizes the plastic as a food source. If it depletes the plastic, it then dies because it doesn't have a food source. Well, I think we're going to probably be able to feed it plenty of plastic, so it will probably live a really long time, sadly. So the last piece with chapter 1 is how do you name microorganisms? Microorganisms are living things, except for viruses. So viruses don't use a genus species. They have like a family name. So their name is a little bit different, but for our other organisms, and again, especially our bacteria, we're going to use a genus species for their names. Genuses are like what we'd equate to our last name. So we share our last name with a lot of different individuals, but our first name's unique, and that's the species. Okay, so with a bacteria, their genus tells you kind of, oh, these organisms have shared characteristics, so they're really similar, but then that species name tells you, like, okay, a little bit more, like, what's their unique characteristic that I could use maybe to identify them. When we abbreviate that, the genus is uppercase, but keep in mind that the species should always be lowercase. So one of the things I like to do is look at the label of like probiotics, and if they have like 2 uppercases, if the species is uppercase, I'm like, clearly scientists weren't involved in this, and they don't know what they're doing. And sometimes I actually contact the company. They usually politely say, "Thank you, crazy lady," but social media doesn't treat me quite as kindly. Sometimes microbes are named after the scientists who identified them first. So E. coli is one that's actually, the E is the last name of the scientist, the doctor who first identified and shared the characteristics of it. In other cases, we can use the shape or the color of it to give us our name. So I'm going to go to the next couple slides to show you some examples of different shapes and arrangements, and then we'll talk about how that fits in with the naming. All right, so you see on this slide, we have some different shapes and some different arrangements. So you'll notice if you look on the illustration column, you can see that we have 2 different types of shapes. So we have circles and we have these rods. Okay, so the circles are known as cocci, coccus for 1, cocci for 2. So they can bind together. So on their outside surfaces, they'll actually interact with each other, and so they'll kind of be like stuck together. So they can form different arrangements. So that's how many of them come together and what shape do they form when they come together. The rods, I always think they look like hot dogs. So these are known as bacillus or bacilli. There's more of them. Okay. So the arrangement again is how they come together, and so you can see you can have 2 of them together, and so those would be known as diplococcus. Diplo referring to the fact that they're a pair. If we have 2 pairs coming together for a group of 4, those would be known as a tetrad. Now we can have a chain of them, almost like a pearl necklace, and that would be known as streptococcus, if those are spears, right? So if you get strep throat, this is streptococcus, usually group A infection, and so your doctor might do a fast test in the office, and the bacteria has this antigen that reacts to the test, and so you get a positive result. In some cases, they would send that out for culturing and then look at it underneath a microscope, and when the scientists looked at it underneath the microscope, they would see that you have this chain, this pearl necklace that's showing up. So never throughout the course in the lab, you'll get to see streptococcus, you'll get to see tetrads, probably see diploids too. The other arrangement that you'll get to see is these clusters. And there's not like a set number in the cluster, so you could have like a cluster of 3, you could have a cluster of 5, 8. I just don't want to pick all odd numbers. So just all these irregular clusters, kind of like if you're at the grocery store and you're in the produce aisle and you're getting grapes and you're cut those grapes in those bunches. Some bunches might have 3, some bunches might have, more, but there's no like set number when you cut them. So those are known as staphylococcus. So staph means grape-like. So again, they look like grapes if you look at them underneath the microscope, and these clusters are very much like grapes where you have not a set number in the cluster. Okay, so if you've heard of Staphylococcus epidermis, Staphylococcus aureus, S. aureus, these would all look the same underneath the microscope. What they look like when they're growing on a plate is different. So shape and arrangement is what you see under the microscope. When we look at what it looks like growing on a plate, it can look different. So you wouldn't be able to determine its shape and arrangement when it's growing on like a nutrient agar plate. Okay. So now with bacillus, a lot of those are just single rods, but we can also get streptobacillus where those are chains. So just like streptococcus, but they are a chain of rods. So there is a genus Streptobacillus. There's also genuses. There's a genus Bacillus. So just see where. So definitely during lab you'll get to see these different shapes and arrangements, but you should know these ones. Do be aware that there are other types of shapes. So there's one that's like a comma shape. That's a vibrio. I was actually just reading this article about bacteria that look like stars. So I can post the link to that article. So there's lots of different shapes, but the dominant ones that you're going to be seeing are cocci, spears, and bacillus rods. Okay, so now we're looking at chapter 3. And chapter 3 introduces you some basic kind of lab techniques, some of which you'll be doing this semester. So I just want to go over the 5 eyes. So in the original uploaded slides, I had additional slides. I'm not going to cover those because those techniques are going to get covered as we'll do them in lab. I just want to touch upon the 5 eyes from the textbook. Okay, so our 5 eyes are inoculation, incubation, isolation, inspection, and identification. So in order, pretty much anytime you're working in the lab, you get a sample, you're going to inoculate that into media. So there's different types of media, but you would transfer that in. You would then incubate it, and incubating is making sure you set the conditions so it's close to its natural environment so that it would promote growth. So the media you inoculate is going to give the nutrients, but we want to make sure that all that the other environmental factors, which we'll talk about in a moment, are controlled in what it needs to be. This allows you to isolate. So in some cases, you might have multiple different bacteria present, so further isolation might be done, or a subculture might be done, so that you have a pure culture. You only have one species of bacteria, because when you go to inspect it, you want to make sure that you're not inspecting this mishmash of stuff, that you're looking at one species, so that when you're looking at those characteristics, either under the microscope, looking for the shape and arrangement, or if you're looking at what does it look like on the agar, and you're trying to eventually identify it, that you're not looking at a whole bunch of different bacteria, that you're only looking at a specific one. Okay, so inspection would be this like visual looking at it. And then identification, this is where you could do additional biochemical tests. So some of those you'll learn about this semester. Some of those we'll talk about when we talk about metabolism. But things like what sugars does it metabolize? Does it produce acid? Does it not? Does salt inhibit its growth? Can it grow? We have agar plates that are made up of blood, and so can it grow on that? All these different things help us to further identify what it is. Okay, as I said, with inoculation, you're adding a small sample. So you'll use an inoculating loop, or also there's a needle. Is usually the method in which you pick up their sample and then transfer it into the media. And there's several different ways to transfer, and you're going to be learning that this week in lab. We then put that media, whether it's a plate or whether it's a liquid in a bottle, in industry where they're making vaccines. They're actually, a lot of vaccines are made of chicken egg, so they would have an incubator that they put the chicken eggs in. Okay. With incubation, we want to make sure we keep that sample at a set temperature. So most of, if we're thinking about industry and whatnot, usually they're trying to grow something that can grow in humans. So it's going to be our body temperature that they're going to set it at. So that's going to be like 37 degrees Celsius, okay, because that's what our body temperature is. So it's not just temperature, it's things like atmospheric gas. So does this microorganism like oxygen? Does it not like oxygen? Does it like CO2? Does it need more nitrogen present? So if we think about all the different areas in which a microorganism could grow in, if it grows in our lung, our lung has this inhale and this exhale, and so we have this constant changing of oxygen and carbon dioxide amount. If we think about deep in our tissues, if the microorganism likes to live deep in tissue, then it probably likes a lower oxygen concentration. So we need to make sure that we provide that for it. If it's a microorganism that grows on our skin's directly in contact with the atmosphere, so it's going to have a higher oxygen content. Now, if we were doing an environmental sample, things like temperature, things like gas, and nutritional needs are going to change, and so we want to make sure that we're providing that. So some of the soil bacteria, if you cultured at room temperature, 22 degrees Celsius versus 60 degrees Celsius, you're going to get different microbes growing. 60 degrees Celsius is, it's like, the sun beating down on soil. Like, it's a much warmer temperature, and so it's not surprising you get different growth. And so again, incubation, what are the conditions? That's important to keep in mind. Okay. We know that the microorganism is growing when the media becomes cloudy. So originally the media should be clear, like you could be able to see through it, but as the bacteria grow and increase in number, the media actually clouds, and that's because you're looking at all these individual cells spread throughout the media. Right, so we need to make sure that we're isolating so that we have a. So one of the ways in order to get a isolated colony, a pure sample, is to do a streak plate method. And so this just allows you to dilute out the sample as you move across the plate. This is a technique that you'll learn in lab. So I'm not going to go over it in more detail because it's really tough to show you how to do it without like being in front of you and demoing it. Another technique you'll get to do is the spread plate method, and this allows you to look at kind of like the concentration of what's present, and this is another technique you'll get to do during this semester, and another one that is easier to understand if you have a demo to see. So then lastly, with inspection and identification, you're going to do any additional test, biochemical test or genetic analysis, or you could do it like an ELISA to determine what organism you have. So certain organisms are going to express certain proteins, so an ELISA might be used to detect that protein. Genetic analysis would look at the genome, and is this gene present? Is there a gene for antibiotic resistance? Well, only this strain, only this species has that gene, so that's the gene it is. Biochemical test, usually you have to do multiple tests in order to determine what you have. But in many cases, purchasing of the media and making of the media is a lot cheaper. So biochemical tests are kind of the go-to in identification. Genetic analysis and immunological testing would cost more. So in some labs they're going to opt to do the biochemical test. And you'll learn how to do the biochemical test. Sometimes I do a mock lab where you get genetic analysis for your organism. So we might do that later on in the semester as an in-class